So we're going to talk about the heterogeneity of autism, and I don't know if people have talked about this earlier today, but here's just a Venn diagram showing you the features of autism. And in these blue circles here are the core clinical features of autism, which I'm sure you all know about: social communication deficits, repetitive behaviors, and then language disabilities. But surrounding these three features are a great deal of comorbidities and coexisting features that make autism a much more complicated disorder. So we have intellectual disability. In addition, there are these symptoms of irritability going on in children. Layered onto that are anxiety, hyperactivity, mood disorders, sleep problems. And then there's medical disorders as well, such as seizures, gastrointestinal disorders, and immune dysfunction. And so it's true, when you see a child with autism, their behavioral phenotype can vary significantly from very verbal and high functioning to very low functioning, but I'd like to emphasize that biologically they're also very, very heterogeneous, and that there can be comorbid features that need to be treated separately from the autism. And then the heterogeneity goes beyond just these comorbid features, the genetics of autism. Here's just a slide showing you all of the different chromosomes that we have. And basically, there have been genes on almost every single chromosome that have been implicated in autism. So it's an incredibly complicated disorder that is definitely going to have more than one single cause or etiology. Likewise, the neuroanatomy of autism, it's hard to actually, so in this figure, I've outlined some of the brain regions that may be associated with the core clinical features of autism as defined by the DSM-4. And this is probably not a complete list of the brain regions that may be impacted in autism, but it's illustrative of the fact that it's not like Alzheimer's disease where there's one structure that we know is very, very impacted. The neuropathology of autism is going to include networks of brain regions and the connectivity between these brain regions. And so it's, again, a very complicated and heterogeneous disorder. And what I'll show you a little bit later on is the data from the Autism Phenome Project is showing that there's not just one neural pattern of abnormality in autism. There's going to be multiple neural phenotypes as well that may be associated with some different immunological and genetic profiles. So we're really not dealing with one disorder here. So this is just to reiterate that point. What causes autism? There are likely to be many different causes of autism and many different types of autism. And there was a study published a few years ago by Dan Geschwin where he coined the term autisms, not autism. And I think that has reached the lay public. People talk about that all the time now, that there are autisms. And here's just a simple diagram of what could arise to these different subtypes. You have multiple genes. I just told you there's genes on almost every chromosome that have been implicated in autism. And then the environmental factors that Irva was talking to you about today. And some mix of those is going to lead to different types of autisms with different underlying biologies that could potentially require different treatments or have a different prognosis. So that's where the Autism Phenome Project comes in. This was really the brainchild of David Amaral, who's the research director at the Mind Institute. And in 2004, they started planning for the study. And he really wanted to define the different biological phenotypes of autism. And so the Mind Institute was a very fortunate place because we have faculty from many, many different disciplines, both in the clinical realm with excellent behavioral psychologists and developmental psychologists, but also we have immunologists, geneticists, brain imagers like myself, and a host of trainees and graduate students. I was a postdoc when I came onto this project that were willing to spend many, many hours collecting data for this project. So I will spend the rest of the talk talking to you about the Autism Phenome Project and some of the data that has been published and that has come out of the study. So a little bit more about the Autism Phenome Project. The vision of this project was that it would be a very large-scale study. We currently have 300 to 350 families enrolled. Ideally, this would be a study of thousands of individuals, because when we're talking about different biological phenotypes of autism, we can be talking about small subgroups. So Kathy was saying in the talk before that 2% of individuals with autism have Fragile X. Well, if you have a sample size of 100 kids and only 2 of those kids have Fragile X, it's hard to do science, to collect data and to analyze the data on those 2 kids. And so we wanted to have a large-scale study. Like I said, initially we wanted this to be a multi-center study, but it just became too difficult and costly to do that. So we're currently at the Autism Phenome Project, and in some ways, in many ways, this is like a pilot project for what should be done in the future with thousands of children. Children are recruited between the ages of 2 and 3.5 years. We wanted to get children as early as possible after clinical diagnosis. One of the reasons for that is because we didn't want children to be on a lot of medications or to have had a lot of intervention already coming into the study. We also were focused on brain development, and brain development is incredibly dynamic prior to 5 years of life. And there have been very few studies that have focused on children this young in autism using neuroimaging for reasons I'll talk about in a second. But that was another reason why we wanted to target this very focused age group. Our study includes all children with ASD with very few exclusions. Many of the published studies that you see are high-functioning individuals or boys only, or those that don't have any neurologic comorbidities like seizures or Fragile X. We wanted to include all of them because we wanted to study all of them. And that was very novel, I think. In fact, we've had papers rejected for this reason, which I think is just a reflection of where the field still is as far as thinking about very homogeneous groups of autism. So again, we have both boys and girls included. And we recruited age-matched, typically developing children to serve as controls for this study. It's also novel because this is a longitudinal design. So children come into us at 2 to 3 years of age, but we actually follow them on an annual basis for 2 additional time points currently. Their second time point, they receive another MRI scan, and the third time point, we do the whole battery again, basically, in them. And this is aimed at looking to understand the trajectories of development, because of course at 2, children aren't doing very much yet. But by age 5 and beyond, we hope to actually continue the Autism Phenome Project as long as these people will let us follow them. Currently we're trying to follow these children into middle childhood. But knowing the course of the brain development over time is very important, as well as the outcomes of these individuals. How do they end up in school? Are they able to attend mainstream school? And what are the predictors of those things? That's really what we're trying to get at with the Autism Phenome Project. And again, the aim is to identify different subgroups of autism. So what do we put these families through? Well, it's a very intensive study. But again, we've had over 300 families participate, mostly from California, but we have had participants come from as far away as New York and Florida, because they've heard about the study and want to be involved. So the children come in. It's actually a family study because we collect a lot of information on the children, but we also involve the parents very much as collaborators to get all of this data in their children. We actually also draw blood from the parents so that we can get genetic information. So they get a battery of diagnostic confirmation, cognitive testing, language assessment, and then Kathy is actually directing the medical component of this study, where we do a medical exam, we collect medical histories on all of these children, and the blood draw part. From the blood, we get information about immunological function as well as genetics. And then the nighttime MRI is where I come in, where we're getting structural MRI scans, structural and functional MRI scans in these children to look at their brain structure and function. And then we actually also have a visit where they're doing an EEG study. And so this is the cap that people wear that have all of the electrodes and is looking specifically at temporal dynamics of brain function. So you can appreciate that we ask these families to come in for 5 different visits. One of them is a nighttime MRI scan. We draw blood from all of the family, and then we ask them to do it all again in the next year and the next year. So we have a really dedicated group of families participating in this study. And again, it's a treasure trove of data. There really is no other collection, no other data set that has data like this. And in some ways, we've just started mining this data to really know what's going to come out of it. So let me just talk to you a little bit about how we do these MRI scans in children, because this is very near and dear to my heart. This is actually what I was recruited to the Mind Institute to do. David Amaral sat me down in his office and said, okay, you want to work in my lab? Figure out how to scan a 2-year-old with autism without using sedation or anesthesia. And I thought, this is going to be the shortest postdoc ever, because there's no way I'm going to be able to do this. But here I am, many, many years later, and we've actually been incredibly successful in doing this. It's not been rocket science by any means, but it has been sort of arts and crafts. So, no, I'm joking. I mean, we've scanned now over 300 children. We've scanned them longitudinally and acquired probably 600 to 700 scans, all during natural sleep. And this is in collaboration with the parents and the child, developing an individualized strategy for every single child, mimicking their bedtime routines at the magnet, to get the children comfortable, trusting us, the parents trusting us to have them fall asleep and be in the scanner. So this is what the scanner looked like when I first got in there. And then in consultation with Sally Rogers and other developmental psychologists at the Mind Institute, this is what we did to make it a little bit more inviting and friendly for the children. We use these blue hangings to cover up any distracting features. We actually cover up the scanner itself. We made a nice big family bed that's covered with really comfy memory foam, and the child can bring their own comfort items and stuffed animals to lie down. They can snuggle up with their parents to help them fall asleep. And this has all been, like I said, it's not rocket science, but this has all been tremendously helpful in our success in being able to get these scans. Prior to our study, there have only been maybe three other groups collecting imaging data in children this young, and they've all required using general anesthesia, which is safe. There's minimal risk, but since ours is a longitudinal study, we didn't want to expose the children to repeat anesthesia, and so we really had to take sedation out of the protocol. And it's worked really well. Our success rate, like I said, is about 90%, so it doesn't work in all kids, but it works in many, many of the kids. And I don't know how many of you have ever had an MRI scan, but they are loud. And I am shocked every single time that a child can sleep through this. My daughter was actually, when I first got into the scanner as a new postdoc, I thought, okay, this really is not going to work. But children can sleep through a lot. So part of what we do, this is my son when he was 12 months old. I am the crazy scientist mom making my children do research. Both of my children have been involved in research at the Mind Institute. Ryan was in the infant sibling study, and Clara, my daughter, who's not shown here, was in the APP as a control. But we have the parents get the children used to wearing headphones at home. They start practicing in the comfort of their own crib or toddler bed, getting used to earplugs and headphones to start to get rid of the sound. And then on the night of the scan, what do I have here? That's a child who's sleeping, and we have lovely research assistants who are there monitoring the child the whole time, making sure they're staying in deep sleep before we start anything. And if the children do wake up during the scan, they pull them out right away. The parents are inside the room on that rocking chair, and they're right there to comfort their child. My daughter did wake up. She did 3 scans for this study, and she did wake up 2 of the times. And I quickly kicked into mom mode instead of researcher mode and freaked out like anybody would. But she was fine. She didn't even remember it because they're in such a deep sleep before we even start the scan that they don't really realize what's happening. By the time they are sort of aware, mom's right there and the noises have stopped. And so it's been a really successful technique. One of the things that is my greatest accomplishment, I think, is that I've had hospitals call me to ask me about this protocols because they're interested in taking sedation out of their own protocols because too many kids are getting sedated for too many MRIs. So that's been really rewarding. And I've also shared these methods with many researchers around the country to the point that now IRBs will not allow you to use sedation for MRI scans in young children anymore. So a victim of our own success, maybe. So let me get on to the science. These are some of the pictures that we have acquired through the study. And what I'm going to talk to you a little bit about is the different measures that we can get from these brain scans. So we can look at brain structure, which is basically measuring the volume of everything in the brain. And we do that with the top row of images. That's called a T1 weighted scan. We can also look at the cortical folding patterns, which is the sulci and gyri are actually folded in in the same general pattern in all individuals, but there is a lot of individual variability, and one of the earlier studies that I worked on showed differences in cortical folding patterns in older children with autism. We can also look at cortical thickness and surface area. I'll also talk to you a little bit about, mostly with the brain structure studies, I'm going to talk to you about volumetric studies because there just isn't enough time to tell you about everything that we do. But I do want to touch on some of the connectivity studies where we're looking at, there's two different ways to look at connectivity. One is to look at the white matter microstructure. So this is the bottom picture there. In these pictures, you can see the white matter is just white. You can't actually tell where those fibers are going. But using a sequence called diffusion-weighted imaging, we can actually then break down the different colors or the different directions that the fibers are going in, and we can actually track fibers going from point A to point B in the brain to see how they're structurally different in kids with autism. There's another type of sequence called the resting state functional connectivity sequence, which I won't be able to talk about today, but that is measuring the actual activation of the brain. So even though the children are sleeping, their brains are active. There's a very wide area of research now on so-called resting state networks. Your brain is active all the time. We're also doing studies sort of combining the structure and connectivity. So if we find a structure that's abnormally large, we then want to look at, well, does this matter? And we look at the activity of that structure and how it's connected to other brain regions to see if the activation is also different. So I'm going to focus the talk on three different neural phenotypes of autism. One is going to be this notion of early brain overgrowth. That is probably the most consistently found result in MRI studies of autism. And this is, well, I'll get into that. The second is abnormal amygdala enlargement, and I'll tell you about the amygdala in a few minutes. And then white matter connectivity. So this picture here is now actually tracking fibers that go through the corpus callosum, which is the largest fiber bundle in the brain. This comes from diffusion-weighted imaging. So first, let me talk to you about the abnormal trajectory of brain growth. So I just want to note that Leo Kanner, in his 1943 paper that first described autism, already noted in his paper the presence of large heads or macrocephaly in autism. He noted that physically the children were essentially normal, however 5 had relatively large heads, going all the way back to the first description of the study. And so it's really been from the very beginning that we've been investigating this. The earlier studies looked at head circumference as a proxy for brain size, which you can do up until about age 7. So your head circumference matches up very well with how big your brain is. And what the early studies showed was that here in blue are the normal curves based on the CDC norms, but in children with autism, this is going retrospectively back, this is their age and months plotted here, that sometime in the first year of life, the trajectory in kids with autism diverges and their brains appear to start growing too rapidly. And this was followed up by several MRI studies showing the same phenomenon, but now actually measuring the brain. This is now years on this axis, and what this is showing is that in the younger years, the preschool age studies, there is this 5 to 10% enlargement on average in brain size in children with autism, but that this difference seems to disappear as individuals age. So that's why I call this an abnormal trajectory of brain growth. It's large early on, but sometime in middle childhood, adolescence, this difference seems to go away. And we don't know yet whether it's that typically developing brains catch up, or do people with autism, do their brains diminish in size? We don't know that because we don't have the longitudinal studies yet to track brain growth in the same individual over time. That's one of the goals of the APP. So what we've been doing in the Autism Phenome Project is to look at this phenomenon of early brain overgrowth and to explore the heterogeneity of it. So the first thing that I did was measured everybody's brain and just simply plotted brain volume and compared it in ASD, which is in red here, versus the typically developing children. And we replicated the previous findings that on average, if you just take the average of the children with autism, their brains are about 6% larger, and this is matching for age and things like that. But look at the variability in brain size, and this is actually plotted here. This now is age on the x-axis, and this is brain size. Now I've just plotted each individual child with autism, their brain size, on this graph. The black line is the mean for the typically developing children, and I just want you to notice the spread in this. It's not that all children have larger brains. You know, these are the kids with the largest brains, but there's a lot of kids with average brain size and even kids with smaller brain size. And so what I wanted to do as a very first pass was to start parsing out these different groups to look for biological characteristics that may be explaining or associated with different brain size. So the first thing that we did was to look at behavior. So you know that some children with autism have a regressive onset, or so-called regressive, where they develop relatively normally in the first year of life, but then start to lose skills later on in the second year of life. And then there's children with early onset autism, who the parents report something was different from the very beginning. And what we wanted to look at was there any sort of distinction in brain size between children who had a regressive onset and early onset. And so we looked at the biggest of the big. So this is megalencephaly, which is the brain correlate of macrocephaly. So you guys have probably heard about macrocephaly more commonly. That you call it macrocephaly when you're measuring the head, but you call it megalencephaly when you're measuring the brain. And we looked at the biggest of the big kids here. So this is that same plot that I showed you before, but this time I'm just highlighting the largest brain kids. And what we found was that 88% of the kids with megalencephaly had a regressive onset. In fact, this is a child, these are all matched for age, these are just 3D reconstructions of their brains, and I think you can tell this kid has a pretty big brain, right? And he has a regressive onset. What we found was that this abnormal brain enlargement actually was only present in children with a regressive onset. The children with early onset autism, their brain sizes were actually pretty normal. This was actually counter to our original hypothesis, because you would think if autism was there from the very beginning, autism is there from the very beginning, even in children with regression, but we predicted that we would see larger brains in the early onset kids, and so this was a surprise finding to us. But it is, I think, very intriguing, because what you can start to do now is to look at head growth trajectories, at your well baby pediatrician visit, and if a child, particularly if there's a familial risk for autism, say they have an older sibling with autism already, if that younger sibling, their head circumference starts to deviate sometime in the first year of life, that would be a red flag to me. That's what we suggested in the paper, to watch this child very closely for any sort of loss of skills that they may have acquired so far. So this was just our first pass into this. There's a lot more to explore with behavioral correlations and brain size, but brain size is it's a sledgehammer approach right now. We're really at the tip of the iceberg. But I'm gonna go on with the brain size story for a little bit more. We're also very interested at the Mind Institute in neuroimmune links with autism. And some of you may have seen recently in the press, a few weeks ago, there was a lot of hoopla about the maternal autoantibody related autism. And so this is the work of Judy Vandewater and Melissa Bauman. And this is, for those of you who haven't heard of it, this is basically Judy's initial study showing that, this was in the CHARGE study, so a very large population-based study, and she identified antibodies in the mother that were only present in mothers who had children with autism. Mothers of typically developing children never showed these antibodies, and these are IgG antibodies that are active during pregnancy. So when during gestation, the fetus does not have its own immune system, it's relying on its mother's immune system. And mothers with this abnormal IgG antibody have children with autism. And so that's the association there. We wanted to look to see what the neural phenotype of these children is, since we do think it's a gestational insult. And what we found here is that, again, these are the children who are on the larger side of brain volume. Here's a plot showing the ASD, what I call IgG, or the maternal antibody-related autism. And their brains are 14% bigger, as opposed to the 6% bigger than typically developing kids. And indeed, they're 8% larger than the rest of children with autism. And so this actually, the really cool thing about this in the Mind Institute was this was replicated in a non-human primate model of this same maternal antibody by Melissa Bauman. And what's exciting is that we have a human phenotype now. We have maternal antibodies and a larger brain size. Then we have an animal model that's showing the same thing. Now we can use the animal model to go in and look at mechanism, something we can't do in children, right? The study that Melissa Bauman did was actually taking these same maternal antibodies from children in the APP, so moms like me, well, my blood I think was in the study, but, and putting it into non-human primates during gestation, during pregnancy, and letting the monkeys grow up and watching their behavior and their brains. And their brains actually are also larger. And so what we can do now is to look at the mechanism. We can look at different points during gestation to pinpoint when this brain growth is going on, this abnormal brain growth is going on, and try to get at underlying etiology. So that's one really exciting component. Again, only possible at the Mind Institute, where you have primate researchers across the hall from neuroimagers working on the same kids and the same antibodies so that we can actually talk to each other. I mean, this happened serendipitously. Melissa walked out of her office and said, hey, I have this cool finding. And I said, well, I have that finding too in my kids. That's pretty cool. So that's been a very exciting avenue of research. And what's exciting about this maternal antibody related autism is that it could potentially be prevented. So the mothers with these identified antibodies could potentially have these bad antibodies removed prior to pregnancy and potentially prevent autism. So there are studies going on right now to look into this. This is only present in about 7% of cases with autism, so it's a sizable subset, but it's certainly not all cases with autism. So let me also talk about, I'm not a geneticist, so I'm being brave by venturing into this, but genetics of abnormal brain enlargement as well. This is from the blood that we collected from the study, and there's something called differential mRNA splicing. So here's my attempt. Here's a gene, and the active parts of the gene are the exons in yellow. And when messenger RNA comes in and splices up the gene to then go on and make protein projects, they can actually splice the gene in different ways. And so there are 3 different patterns of exon splicing here that can go on and cause different gene products. And so we wanted to look, this is with Boriana Stamova and Frank Sharp, who are upstairs from my office at the Mind Institute. They wanted to look at the phenomenon of these large brains versus normal-sized brains in ASD and see if there are any genetic differences between these groups as well. And what they found, she has very fancy figures. I don't understand this plot at all, but this one, you can see. So the large total cerebral volume, that's what that stands for. These are the big brain kids, and these are the normal-sized normal brain size ASD kids. They're split, right? There are different groups of genes that are leading to these different brain sizes. And this paper is under revision right now, and we were actually surprised at how well these groups differentiated with genetics. So this is just an example of, again, the way looking at the heterogeneity of autism by looking at multiple avenues of biological data to try to come to a converging sort of etiology for a subtype of autism. I just wanted to address these smaller brain kids as well, because I've been talking a lot about abnormal brain enlargement. What we found in a study by Christy Rossi in the APP a few years ago is that before I was talking about mothers with antibodies that attach to fetal brain, now I'm actually talking about the autoantibodies in children themselves. When we collect the blood from the children, and then stain it on brain tissue, we actually can identify antibodies that are directed at the child's own neurons or directed at neurons. And this is very early on. We just started looking at this in the APP. We don't have all of the stains done, so we actually don't have all of the children characterized yet. But here, it sort of makes sense that we see that these children who have these antibodies directed at neurons, at their own neurons, are in a group that have smaller brain size. And so this example is just thrown in because I don't want to think that you to think I'm biased and only looking at the big brains. We're also going after the smaller brain phenotype as well. So let me move on to the amygdala. The amygdala in autism has a rich history. People have been talking about it for a long time. This is the amygdala outlined here. It's a structure deep within the medial temporal lobes. And it's involved in fear detection. So when you're going about in a social scene or any sort of scene, say you're a monkey and you come across a snake. It's innate that you will be afraid of that snake, and it's your amygdala that's doing that. Primates who actually have no amygdala because they've been lesioned are not afraid of the snake, and they will pick it up and swing it around by the tail. These are plastic snakes. So how does that lead to autism? This is something that I think about a lot. Well, say you have a child who has an amygdala, but we know that the amygdala is abnormal, at least in its size, and we know that the amygdala is involved in fear detection in your environment. Well, fear could be fight or flight, the snake is going to bite me and I'm going to leave, but it could also be in a social setting and understanding what this person is trying to say or communicate with me. And so I think that it's a more subtle difference in autism. It's not your fear system is not completely turned off. Children with autism still have fear of objects they should be afraid of. But I think that it can also be affecting their social environment as well. If you can't learn in your social environment what you're supposed to be afraid of, you might not be able to navigate very well in your social environment either. And again, I just want to point out in the Lee O'Connor paper, he also described anxiety in children with autism, and the amygdala is tightly linked with anxiety and fear, as you could imagine. So I won't read through those quotes. If you haven't read this paper, it's actually a pretty wonderful paper, and if you're in the field of autism, it's good to know where it all started. So I recommend it. So in typical development, the amygdala grows, and it grows in a linear way, and it gets bigger as it's supposed to. This is across the period of 8 to 18 years of life. And in an earlier study that Cindy Schumann and colleagues carried out in the lab, they looked at amygdala development in this middle childhood time period in autism and typical development. And what they noted, now this pink line still is typical development, and these are 3 different groups of autism. She was looking at low functioning, high functioning, and Asperger's syndrome at the time. This is back in 2004. And you can see that none of the blue or green lines look like the pink line, right? The amygdala is not growing normally. In fact, it's already enlarged by the time the children are 8. In fact, some of these children, by the time they're 8 years old, have amygdalas the same size as an adult, like you or I. And so this is a very dysregulated growth pattern for the amygdala. And so in the APP, the first step we wanted to do was to see if amygdala enlargement was present in 2-year-olds, and it is. So this was an initial paper that we did looking at amygdala size, we outline the amygdala and we look at the size. This is at our time 1 time point at age 2, and then also our next time point at age 4. And what we concluded from this is the volume of the amygdala. This is just the right amygdala here, but it's the same for the left amygdala, is that the red bars are higher than the blue bars. The autism group, on average, has a larger amygdala, and this enlargement seems to be increasing. So now this is just looking at the rate of change. In the typically developing children, their amygdalas are growing at this rate, but the red line is a different slope. It's a steeper slope. So that means their amygdalas are still undergoing abnormal development at this age. What we wanted to do next, which is what I do in the Autism Phenome Project, is to look at subgroups. And so here are the individual trajectories of every child with autism. And what you can see here is that there's a lot of variability in the slopes here. And so what we did was we took this previous graph, this very simple line graph, and we broke it down. We identified 3 different patterns of amygdala growth in children with autism relative to the typically developing. This one group here, their amygdalas got bigger by 20% in 1 year. I had to go back and check my tracings to make sure this was actually real. Interestingly, so this was happening in about 40% of the children with autism. There was another group, though, this was surprising, that had slower amygdala growth than what we would have been expected. And then again, there was a 40% of children who had perfectly normal amygdala growth in this time frame. So what does this mean? Well, what we really want to do now, and what we've just put in a grant proposal to do, is to look at these children into middle childhood to see what their outcomes are. So the amygdala is related to anxiety, Comorbid psychopathology doesn't really show up in 3-year-olds with autism, but it does by the time they reach middle childhood. And what we would predict is that these children with these really abnormally growing amygdalas are going to be the ones who are maybe more prone to anxiety problems later on. And this could be an early neural predictor of those children, and treatments and medications could be given accordingly. And then also to link it to the immune side of the APP, nobody really knows why the amygdala or how the amygdala is getting bigger. And so with the Autism Phenome Project, we have their blood samples. We have Paul Ashwood and Judy Vanderwater to look at the immunological profiles of these children. And he actually found, I had to cheat here and write all the text down because this, again, I'm not an immunologist, but I'm presenting immunology data, that there's a certain type of immune cell, the myeloid dendritic cells that are involved in many different immune processes. They also manufacture and secrete growth factors. And we found that children with autism have an increased frequency, so these are the same children in the APP, have more of these dendritic cells. And we also found an association between the amount of dendritic cells that they have and their amygdala volume. And so this is getting potentially at an immune link to this abnormal amygdala and growth, and again, a better understanding of or we hope someday, a better understanding of the etiology of this disorder. So again, cross-disciplinary research I think is going to be really key. I keep saying that. So let me talk to you very briefly about white matter connectivity and some of the studies that we're doing in diffusion tensor imaging to look at that white matter microstructure. This is a review paper that was published recently, and the only thing I want to point out here is the sample size. So this is a sample size of all of the DTI studies that have been published in autism. And what you can see is most of them have sample sizes of about 20 kids. One study has a sample size of 50 kids. We have 300 kids, and so it's taken us longer to process all of the data, but I think it's also going to be very valuable in looking at different phenotypes and different patterns of connectivity. What I want to focus on here is the study that we've been doing on the corpus callosum, which I showed you this picture before. It's the largest fiber bundle in the brain. It connects your left hemisphere to your right hemisphere, has long distance neocortical projections. And what we did was looked at we looked at where the fibers in the corpus callosum are projecting to. And that's the color code here. So orbital frontal cortex, anterior frontal cortex, superior frontal cortex, and so on. And we looked at the pattern of corpus callosum connectivity. This time we were looking at boys versus girls with autism because we actually have 30 girls with autism. So we wanted to look to see if they have a different neural phenotype from boys. We know that autism affects 5 times more boys than girls, so you would think potentially that they have different underlying etiologies or neuropathologies as well. And indeed, what we found is that there are different patterns of corpus callosum connectivity. This green region projecting to the superior frontal cortex was abnormal in both genders. Both boys and girls had smaller regions connecting to the superior frontal lobes. The frontal lobes are the part of the brain that are involved in everything that makes us human. Planning, decision-making, social interactions. It's a very important and large part of the brain. But what we saw differently was in the orbital frontal region, which has been implicated in social behavior. Only the boys showed this deficit in the size of those fibers projecting there, whereas the girls had a deficit in their anterior frontal fibers. So this is suggesting that there's a different pattern of connectivity in boys and girls with autism, whereas before, people have always just put boys and girls together and looked at a single neuropathology. What this was telling me was that we really need to look at them separately, and we need to study more girls. In our study, which is relatively large, we have 200 boys with autism, but we only have 30 girls with autism. And so another thing that I would like to pursue is to selectively recruit girls so that we can get up to 100 girls with autism and look at their neural phenotypes in detail compared to boys with autism. This last slide that I will show is then we looked at fiber tracts not only in the corpus callosum but throughout the rest of the brain. So we looked at 18 different fiber tracts in the brain, and I'm showing these because these are very pretty pictures. This is done by a postdoc in the lab, Ryan Johnson. And these are the fibers that are affected in boys with autism, and these are the fiber bundles that are affected in girls with autism. And you can tell that they're different. widespread across the entire brain. They seem to have a different pattern of connectivity. And this is really illuminating, right? So we need to look at them separately. Again, that was my point of this slide. So let me just wrap up here. Let me talk to you the take-home points of this talk, what we've learned from the Autism Phenome Project. Brain enlargement. When you hear about abnormal brain enlargement in autism, I would like you to think that it's not all kids with autism who have larger brains. It's actually just a subset of kids with autism. And actually I think one of the things that's happening in the field right now is people are calling into question whether early brain overgrowth actually exists. And it's because they're looking at all of the individuals with autism. It doesn't exist in all individuals with autism. It exists in a subset of individuals with autism. And so I think it's a detriment to the field when people are calling it into question, saying it doesn't exist, because it is a meaningful clinical phenotype. Amygdala enlargement is the same thing. There's different trajectories of growth, and we think that this could potentially predict later anxiety and mood disorders. White matter connectivity studies have shown us that females and males are different. And really the take-home points are just what I said at the beginning, that autism is very heterogeneous, not only behaviorally, but also biologically. Multiple biological subtypes exist. We're really just at the beginning of identifying what these subtypes are. The next step would be to figure out how to treat these different subtypes, and we're not quite there yet, but this is a first step and a first attempt in doing this. And that large-scale interdisciplinary studies are needed to fully understand the causes of autism. And again, with 300 families, the APP is just the beginning. We need studies of thousands of families, but you can see the complexity of this and the cost of this too. This is something that's very difficult to get funded because the NIH likes to put money in safe bets, and this is not a safe bet. This has been called by the NIH a fishing expedition, which, it is in some sense. But there's a place for fishing expeditions too, to generate hypotheses. And so I'll just end with, we have a lot more work to do. We want to look more into the genetics and brain structure. Many of the genes that have been implicated in autism are affecting synaptogenesis, neuronal function, and things like this. I'd like to look more into the immune link with gastrointestinal disorders. That's a very common complaint. This child is, this is when he was 2 and now he's 8. He's come back into our study, and he has a lot of gastrointestinal disorders that have gone untreated because he can't talk and he can't communicate the problems. And so that's something I think we need to look into more. And then finally, do these early neural phenotypes matter? We want to follow these kids into middle childhood and beyond to see if we can predict anything that's happening in the future. So thanks very much. This is my very dedicated MRI team. I can't tell you how many hours we've spent collecting these MRI scans from the 8 p.m. to 2 in the morning. We spent a lot of time together. So we're very close, and it's been a huge team over the years. Some of these people have gone on to medical school or are clinical psychologists now, and it's been rewarding to mentor and see them grow. And then, of course, the rest of the faculty and staff at the Mind Institute who have helped in acquiring all of this data and planning the study, particularly David Amaral, who's not on this slide, but he's on the previous slide, who really developed the Autism Phenome Project.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.